I welcome you all to this video. Good evening, everyone. So we are going to be looking at gametogenesis specifically. We are going to do gametogenesis, of which in this session we are going to cover spermatogenesis as well as oogenesis. So we are going to look at the process of gametogenesis in males, which is classified or termed as spermatogenesis, as well as in female, which is known as oogenesis. So first of all, we are going to start with spermatogenesis. So before we start with spermatogenesis, or rather before we dig into the whole process of gametogenesis, I would like us to look at the site where most of, or rather let me say where the whole process of spermatogenesis is going to be taking place. So we have a site, or rather this diagram that you are seeing here. This diagram, it is a diagram that is the diagram of the testis. So this diagram of the testis, basically it is going to be, or it is rather showing the parts that we are mostly focused on, or that we are mostly interested in. So this part that we are seeing here, as it is labeled, this is the epididymis, and this is going to be the body of the epididymis. Then this is going to be the tail of the epididymis. So as the tail of the epididymis passes, as it has gone passing the other side, we are going to have the formation of what we call the ductus deferens. Then from there, we are seeing also these structures that are appearing like cord-like structures. So these cord-like structures that we are seeing here, they are known as the seminiferous tubules. They are known as the seminiferous tubules. Then I not have labeled here, for example, we have got this structure, this site. At this site, we are going to have a structure that is known as the rete testis in the mediastinum of the testis. We are going to understand more as we are going to do the gross anatomy on these things. So the part that I just want to focus on, or to look at it, is this one. So in the seminiferous tubule, that's where we are going to see the whole process of spermatogenesis taking place. So it is going to be the actual site of sperm production, the same seminiferous tubules. So these seminiferous tubules are just going to be highly coiled tubules that are going to be found in the testis, as you have seen. These are just going to be highly coiled tubules, these that we are seeing here. So the whole process of spermatogenesis, as we have heard, takes place in the seminiferous tubule. So there are certain cells that are found here in the seminiferous tubules that tend to support the same process of spermatogenesis. So these cells, we have cells that are known as the Sertoli cells, and as also what we call the Leydig cells. So I'm going to find that these cells, they'll be found here. So specifically, when you look at the Leydig cells, these Leydig cells are going to be found in between the spaces that are found in these the seminiferous tubules. That's why they are going to find these Leydig cells. So these Leydig cells, they're just going to be acting on the Sertoli cells, stimulating the Sertoli cells, so that the Sertoli cells can now act, or rather bring about their function to support the same process of spermatogenesis. So specifically, these Leydig cells, they are just going to be stimulating spermatogenesis by acting on the Sertoli cells. That's one of their functions. And the other function is that these Sertoli cells, they promote the development of the male secondary sexual characteristics that we all know. So the other cells, which are the Sertoli cells, these cells basically, they are also known as supporting cells, or these cells, they are the cells that are involved in the support of the whole process of spermatogenesis. So you are going to find that they will be involved in nourishment. So they are going to have that function that is going to be involved in the supply of nutrients, and as well as other growth factors to these germ cells that are going to be found in the seminiferous tubules, that are going to give rise to the gametes that are being produced, which are the sex, the male sex cells. Then they are also going to be involved in the process of forming what we call the blood-testis barrier. So this blood-testis barrier, actually, it is formed by the tight junctions that are formed that are in between the same Sertoli cells. So this blood-testis barrier, it creates a barrier that prevents the exposure of immune cells to these germ cells that are dividing to form the sperm cells. So you're going to find that they'll be forming this barrier so that they prevent an immune reaction onto those testes, or rather onto those cells that are being formed from the progenitor, from the same cells that are found in the seminiferous tubule to form the sperm cells. and they are also going to be involved in the process of phagocytosis. So at some point, these Sertoli cells, they actually have got a phagocytotic action, in which they tend to remove those residues that are being shaded by these maturing spermatids. So we're going to find that as these cells inside the seminiferous tubules are dividing to form the spermatids, there are certain structures that are going to be being shaded off by the maturing cells to form now the spermatids. So those structures that are being shed off are going to be phagocytosed by these Sertoli cells so that they can be eliminated. So basically, these are some of the functions that these cells they tend to play. So these cells are going to be found in a seminiferous tubule, and as we have heard, the seminiferous tubule is going to be the site for the whole process of spermatogenesis, or that's where the whole process of sperm formation, or rather spermatocyte formation, is going to be taking place. So having looked at this, we would like now to introduce now the whole process of spermatogenesis. So before we enter into spermatogenesis, first of all, we have to understand the concept, which is the concept of gametogenesis. So gametogenesis is basically a process of forming gametes from the primitive sex cells, or from primitive stem cells. So you're going to find that these primitive, as I've talked about, these primitive sex cells are going to be the one that will be involved in the forming of the gametes. Yeah, I hope we are following. So they are the one that are going to be involved in the formation of these gametes. So these primitive sex cells, or rather let me say the primordial germ cells, they are more like the ancestors of the germ cells. They are the origin for the same gametes that are formed. So these cells are going to be the earliest cells in the embryo that are going to give rise to all future gametes that are going to be formed. They are known as the primordial germ cells. So the whole process of gametogenesis is just the process of forming gametes from these primitive sex cells, or the primordial germ cells. So we all know that the whole process of gametogenesis takes place from the seminiferous tubules in the testes, as we have given a brief explanation on that. So we are going to find that the process of gametogenesis, or the process of the formation of gametes in males, it is what is known as spermatogenesis. Then in that of female, it is what is known as oogenesis. So we have got spermatogenesis, which is specifically for males, spermatogenesis, in which these sperm cells are going to be formed in males. Then you are also going to have oogenesis. So this whole thing, the spermatogenesis or genesis, it falls under the whole concept of gametogenesis, because it is just separated because you are looking at two different people with different sex, with just different sex here, as you have seen. So let us now dig deep into spermatogenesis. So spermatogenesis is actually the process by which these primordial germ cells, they form what we call the spermatogonia cells in the seminiferous tubules. So we are going to find that these spermatogonia in the seminiferous tubules are going to be dividing, or they are going to be developing to form now what we call mature spermatozoa. So this whole process of spermatogenesis that have been summarized on this table here, it takes place in the testes, specifically in the seminiferous tubules, and we are going to find that the cells that we talked about, like the Sertoli cells, they'll be giving some support to the developing cells, like we saw the nourishment, the phagocytosis, and also the formation of the blood testis barrier. Then we are also going to find that other cells, like the Leydig cells, are also going to give some sort of support, as they are going to be stimulating these Sertoli cells and also other functions that are involved in to support the whole process of spermatogenesis. So the whole process of spermatogenesis that we are seeing on this diagram here, it has got 3 stages. So it happens in 3 stages. So the first stage, we are going to have what we call spermatocytosis. That is going to be the first stage. So the first stage, if I may write here, So the first stage, we are going to have what we call spermatocytosis. That is going to be the first stage. Then the second stage is going to be meiosis. And I believe we are all familiar with this concept of meiosis. Then the third stage is going to be spermiogenesis. So spermatocytosis, meiosis, as well as spermiogenesis, these are going to be the stages that are involved in spermatogenesis, in which we are going to see the spermatogonia and the seminiferous tubules maturing into a spermatozoa. So now let's dig deep into this whole process so that we can make use of the diagram that we are seeing here. We try to put some flesh on it so that it makes sense. So the process of spermatogenesis, as we have said, that it has got these three stages. The first stage, it is what we call the spermatocytosis. So under the first stage, which is the spermatocytosis, this stage you are going to find that these spermatogonia cells that are already being formed, they are going to be converted into what we call the primary oocyte. So this first cell that we are seeing here with a diploid number of chromosomes, which is 46 chromosome, of which we all know. This cell here, it is known as the oogonia cell, or rather the spermatogonia. Yeah. So this spermatogonia, basically, it is going to be converted into a primary oocyte in this whole process as we are going to be going down. So starting with spermatocytosis, we said that spermatocytosis is actually a stage in which the spermatogonia cell will be converted into a primary oocyte, and this whole process of converting this spermatogonia into a primary oocyte that we are seeing at this stage takes about 16 days. So from the spermatogonia cell, as we have seen, there's going to be what we call mitotic division, in which this spermatogonia cell, it is going to divide into two daughter cells, which are going to be genetically identical to the mother cell, as we have seen. So when this spermatogonia cell divides into two, it is going to divide into two types of spermatogonia cells. So we are going to have type A spermatogonia cell and type B spermatogonia cell. So this type A spermatogonia cell, it is going to be reserved. It is going to be that cell that is going to be given that special treatment. Say, no, you won't undergo the other side, but we are going to keep you so that we get to remain with a copy of one cell that will be recycled, so that we can have the formation of more spermatocytes. So you are going to find that this type A spermatogonia is going to be reserved, and it will start undergoing mitosis or mitotic division. And when it goes through mitosis, it is going to be providing more and more spermatogonia cells that are going to be formed. As we have all known to say, this spermatocytosis, or the process of spermatogenesis, takes place it takes place frequently, and always it is always taking place. There is formation of this. This is because the reserved cell, which is the spermatogonia cell, it is undergoing mitosis to form other cells, which we are looking at right now. So from the spermatogonia cell, as we have said, the type A is going to be formed and the type B is going to be formed. So this type A is going to be reserved, and the type B is the one now that is not going to undergo the whole process to enter now into meiosis. Then there will be formation of the secondary spermatocyte. So this type B, what will happen is that this type B will undergo division, or rather it will enter into a second stage, which is known as meiosis. So for spermatocytosis, what you just have to know here is that the spermatogonia cell, which is here, it is going to be converted into a primary oocyte via the whole process of undergoing meiosis. But what will happen is that first the spermatogonia cell will divide into type A and type B spermatogonia cells. Then the type A, it is going to be reserved, and it will undergo the whole mitosis cycle so that it can get to form more spermatogonia cells. Then the type B, it is the one that is going to be released to enter into the next stage, which is meiosis. So you are going to find that under this process, as you have seen, the chromosomal number, it is just maintained as diploid or diploid. it is just maintained the way it is. There is no reduction, because the whole process that is taking place here, it is mitosis. Nothing is being reduced. So you're going to find that as the whole process takes place here, this type B spermatogonia is going to be released to enter into meiosis, which is the second stage. So when it enters into meiosis, which is the second stage here, you're going to find that this type B will undergo division, in which it is going to be converted into a primary spermatocyte. So when it is converted into that primary spermatocyte, this primary spermatocyte, now it is now going to undergo the same meiosis 1. So what is happening here is that the type B, it is the one that is referred to as the primary spermatocyte, just to clarify that point. So this type B, as it is released, it will enter meiosis 1. So as it enters into that meiosis 1, it is now going to undergo division, and we know that meiotic division will cause the reduction in the chromosomal number. So you're going to find that the chromosomal number will reduce from diploid to haploid. Half of what we are having here is what is going to be found into the daughter cells that are going to be formed. So you're going to find that there will be reduction in the chromosomal number under meiotic division, and this whole process actually takes about 24 hours just to take place. So you're going to find that as this whole process of division takes place, there's going to be formation of what we call secondary spermatocyte. So these that we are seeing here are what is going to be termed as a secondary spermatocyte. So when these secondary spermatocytes are formed, each is going to be having a haploid number of chromosomes, which means it's going to be having half number of the chromosomes. Then these secondary spermatocytes are going to divide to form what we call the spermatids as they enter into the second meiotic division. So as they enter into a second meiotic division, these secondary spermatocytes, as we have seen here, they are going to divide to form what we call spermatids. So these spermatids are just going to be some sort of immature sperm cells that are not yet ready. So they have to undergo some processes so that they can become ready. So the whole process of forming from this spermatocytes will form spermatids. Then the spermatid will differentiate to form the spermatogoa. It is actually known as spermatogenesis. So these spermatids will undergo a process known as spermatogenesis, which is going to be the last stage, in which they are going to differentiate into a mature spermatozoa cell. This mature spermatozoa cell, it is the cell now that is going to be required for all the process that it is needed for. So the whole process of spermatogenesis will occur when these spermatids, they undergo differentiation and they mature into the spermatozoa cells. So this spermatozoa, the formation of the same spermatozoa actually takes about 24 hours, and this whole process involves what we call metamorphosis, which is going to be the changes in the structure of these spermatids that are formed. So these spermatids are going to change in their structure, and as they change in their structure, they are going to form a more competent cell that is going to be released to say, yeah, you can now go into the world and preach the word. So what we are going to see here is that these cells that we are seeing here, let's try to zoom them so that we know what is happening. This cell that we are seeing here, that cell that we are seeing as the spermatid, this cell that we are seeing as a spermatid, we said that it was having haploid number of chromosomes. So meaning the number of chromosomes had to reduce. So haploid is going to be about 23, of which you know that we are going to have 22 plus the X, the sex chromosome, or the chromosome that will determine the gender of that particular child or embryo that is going to be formed as the fertilization occurs. So it is going to be having a haploid number. So when that happens, this spermatid is formed. It will start undergoing the whole process of maturation. So as a spermatid is formed, it is now going to start undergoing the process of maturation. So what happens here is that the spermatid will turn into a mature spermatozoa through some events that will take place. So these some events will involve changing of its structure, as well as some its intracellular components are going to change, that will form something that is going to support the whole function, the whole process of the activity of the sperm cell that is required for the process of fertilization. So you're going to find that this cell that we are seeing here, it is going to have its cellular content. So we are going to have the content. Let's say this is going to be the Golgi apparatus. Then this is going to be our nucleus. Then this is going to be the mitochondria. Then these are known as the proximal centrioles. We know the centriole and their functions. And also, these are going to be the distal centrioles. So, these intracellular structures, as this spermatozoa is undergoing maturation, or spermatid is undergoing maturation into a mobile spermatozoa, these cellular contents, they tend to change. They tend to the whole structure tends to rearrange itself so that it can now form a competent spermatozoa that is going to be motile and that is going to be ready to carry out the whole process of fertilization. So I'm going to find that the Golgi apparatus there, it is going to be differentiated, or it is going to change, and as it changes, it is going to cause the formation of what we call the acrosomal cap. So this that we are seeing here, it is known as the acrosomal cap. Acrosomal cap, or in some books you find it being written the acrosomic cap. Then the nucleus here, it is what is going to form the same nucleus that we are seeing here. That one, it just remains as a nucleus because it's the one that carries the whole genetic material, so it just has to remain like that. Then the mitochondria will be found at this intermediate or the middle segment. So this O segment here, it is known as the middle segment. So the mitochondria is what is going to differentiate to, it is what is going to be relocated, and it is going to find itself at the middle segment. Then these, the distal centrioles, they will be at this spot. Then the proximal centrioles are the ones that are going to be found at just this spot at the neck. So this part here, it is known as the neck. Then this part, it is going to be the middle segment. Then this part, it is just going to go as the tail. Then the tail here, we know that the cell inside has got some cytoskeletons that facilitate some intracellular processes and so on and so forth. So I'm going to find that this is going to be called the axofilament. So this axofilament will be part of the tail, and it will be facilitating the whole process of locomotion, or the whole process of movement for the same sperm cell. So in a brief explanation, or rather just to go through the same whole process of spermatogenesis, they say that the whole process of spermatogenesis, actually, it involves the spermatogonia, which are found in the seminiferous tubules, to develop into a mature spermatozoa. And we said that this process takes place in the seminiferous tubules that are found in the testes, as it is being supported by cells which are known as the Sertoli cells, as well as the Leydig cells, of which their functions we highlighted on. Then we said that the spermatogenesis process has got 3 stages. There are 3 stages in which it occurs. So we have got the first stage, which is the spermatocytosis, of which is said that the spermatocytosis is just the stage in which these spermatogonial cells are going to be converted into a primary spermatocytes. And as it is converted into that, what happens first is that it is going to divide into type A and type B, of which the type A is going to be reserved so that it can continue to undergo the same process of mitosis, and the type B is the one that is going to be released to enter into the meiotic division. So as it enters into the meiotic division here, you're going to find that it is now going to be termed as the primary spermatocyte. So in that first meiotic division, it is going to divide to form what we call the secondary spermatocytes, and these secondary spermatocytes are going to divide further as they enter into the second phase of meiosis to form what we call the spermatids. Then these spermatids, they will be having what we call a reduced number of chromosomes to half, which is termed as the haploid. And they are going to undergo spermatogenesis, in which they are going to be converted into a mature spermatozoa. So this whole process of spermatogenesis caused this cell now to differentiate. Its morphology is going to change. There are going to be some relocation inside on which its intracellular organelles are going to be rearranged to form a supportive structures that will facilitate the processes that this spermatozoa is actually involved in. So now, in as much as this old spermatozoa cell has been formed and it looks mature, like now is the dude is about to go, it has to undergo some sort of maturation and capacitation. So the spermatozoa undergoes some sort of maturation. So as we looked at this structure here where we said that in the seminiferous tubule here, that's where the whole process is going to be taking place. So when this cell is now formed, the spermatozoa is now formed and it is at this stage, it is now going to be pushed. It is going to move from the seminiferous tubule and it will enter into the epididymis. There in the epididymis, that's where it is going to undergo some further maturation. So there are going to be some sort of maturation, or there is going to be further maturation that will occur to this spermatozoa cell so that it can now acquire some motility and become fully motile after ejaculation. So you are going to find that it will end as it enters into the genital tract. That's why now it will acquire the ability to fertilize an ovum. But at first, this spermatozoa, when it is formed in this seminiferous tubule, it is going to be pushed to the epididymis. There in the epididymis, it is going to undergo further maturation. So this maturation that we are talking about is just a condition in which it will acquire some motility ability so that it can be fully motile. It is just going to be charged up, packaged nicely, so that as it is going to be ejaculated into the female genital tract, it will be able to move, or it will acquire that ability to move, and also it will be ready to carry out the whole process of fertilization. So we are going to find as this maturation is occurring in the epididymis, the final stage of this maturation that tends to occur as this spermatozoa is being prepared, it is known as capacitation. So even though capacitation does not occur into the epididymis, it is also part of the process of maturation for the spermatozoa so that it can acquire the full ability to fertilize the egg. So we're going to find that capacitation will be now the final maturation of the spermatozoa. So what will happen is that when this spermatozoa guy has been released into the female genital tract, it will enter. So as it enters there, it is going to undergo the same process of capacitation in which there are some glycoproteins that are going to be removed. Then there are also going to be some alteration on its membrane. It is also going to be charged to get the full ability so that it can also cause the release of enzymes from the acrosomal cap, because this acrosomal cap has got the ability to release certain enzymes that causes the destruction, or rather the degradation of what we call the zona pellucida, as well as other components of the ovum, so that it can create a way for the fusion of this genetic material here in the nucleus of this spermatozoa, and as well as that of the ovum. So we're going to find that it has to be prepared for those activities. So in the process of preparing it for those activities, it is gaining now the full ability to fertilize the egg as well as to penetrate the ovum. So this whole process of capacitation, as I have highlighted, it takes place in the uterus and also the uterine tube, which are also known as the fallopian tubes. So maturation basically it just involves this guy to undergo more maturation so that it can acquire full motile ability when it is ejaculated. That is, if it is in the epididymis. So when it is ejaculated, goes into the uterus or the fallopian tube, there it has to undergo capacitation. So capacitation will be the final stage of maturation in which this now spermatozoa cell, it has to undergo some processes that will make it to be capable of penetrating and fertilizing the ovum. So these are just the stages, and these are some of the points that we'd like to highlight on this process. More points, you are going to find them in the notes. Then the other part which is the last part of this lecture, or rather of this session, it is going to be oogenesis. It is going to be oogenesis. So oogenesis basically, it is the process by which the oogonia, or rather the primitive germ cells, of which we say that these primitive germ cells are the ancestral cells. They're more like the ancestors where all those gametes comes from. So the oogenesis is just a process by which these primordial germ cells, they develop into mature ovum or into mature oocytes. The ovum are also known as the oocyte. Yeah, the ovum are also known as the oocyte. So oogenesis basically takes place in the ovaries. So I'm going to find that inside the ovaries, that's where the whole process of oogenesis will be taking place. And this process of oogenesis actually has got 3 stages that it follows. First of all, there is the multiplication stage, then the growth stage, then now it has to undergo the maturation stage. So this maturation stage, it is also the stage that is going to be involving the meiosis part. So it has to undergo meiosis so that it can become fully matured and support the whole process that it needs to, or rather it facilitates the occurrence of the whole mechanism that it needs to perform. Okay, so now this is the whole process of oogenesis that has been summarized in this table that you are seeing here, or rather in this diagram. So what happens under oogenesis, as we have heard, is just the formation of the female gametes from the primordial germ cells. So this whole process actually takes place in 3 stages. So the first stage, it is the multiplication stage. So this multiplication stage involves the primordial germ cells to divide by mitosis to form the oogonia. So this oogonia, it is going to be a cell that will be formed from the primordial germ cells. That is the multiplication phase. So this multiplication stage will involve these primordial germ cells, the ancestral cells that are already found there, which are the origin of the gamete cells. They are going to start undergoing multiplication, which is mitosis, so that they can form the oogonium. So as this oogonium is formed, at first, as we have seen the indication here, it is going to be having 46 chromosomes, or it is going to be having the full number of chromosomes that is required, and this will undergo multiple, or rather rapid multiplication, as we are going to be looking at it further as we go down. So I'm going to find that at this stage, it will now, after multiplication has occurred and everything, it will start now undergoing some preparation in which it will start growing and everything and so on. So that preparation stage is what will support it to undergo the whole process of meiosis. So the first stage, it is the multiplication phase, which will involve now the primordial germ cells to multiply so that they can form the oogonium. Then the second stage is going to be the growth stage. That growth stage, as we have talked about it briefly, we are saying that it is now going to involve the growth in the size of this oogonia, and it is going to facilitate the growth of the cell, in which it is going to have increase in the intracellular organelles to support its process that it is undergoing. Then the RNA is also going to be needed to perform its early embryonic development, and also some nutrients are going to be needed, so there will be accumulation of nutrients to support the whole process. So I'm going to find that under the growth stage, this oogonia cell, it is going to be surrounded by certain types of cells that are known as the flat follicular cells. These flat follicular cells are the ones that are involved in the follicular development, or rather they form what we call the primordial follicle. These primordial follicles, they are the one that tends to cover the developing ovum. So as the ovum is developing, growing and going and so on and so forth, it is going to be taking place inside this primordial follicle, as it also undergoes its own stages from primordial follicle, primary follicle, secondary, then until it reaches the whole process of maturation, just like the way we have seen. So we are going to have the follicular development that will be happening simultaneously with the process of oogenesis. So first of all, we are going to talk about this. We look at what is happening to this ovum. Then now we are going to combine this. We are going to put it here to see now how these 2 things are happening together, because we need to understand those things. Okay, so now the last stage is the whole stage of meiosis, or meiotic stage that we are now going to start entering into. So at first we said they have to undergo multiplication phase in which the primordial germ cells, they divide into a mature ovum. Then they will have to undergo growth, in which that cell has to grow to prepare itself to enter into the meiotic stage, which is the maturation stage. So now let's dig deep into the meiotic stage. Okay, so as we are seeing from the diagram here, the primary oocyte undergoes the process of mitosis. So what happens here is that this whole process here takes place before birth. Yeah, first before we go down here, it takes place before birth. So we're going to find that the child that is going to be born, or the baby that is going to be born, will be born with all the ovum, or rather the eggs that is required for her lifetime, for her lifetime of reproduction. Yeah, she's going to be born with them. She's not going to be like males who have got already the reserved type A cell that undergoes mitosis every time. So you're going to find that that's why they tend to reproduce. They don't undergo the stage of menopause because of the reserved type A cell that we talked about. So under oogenesis, so we're going to find that this cell will enter now into the meiotic division. So we're going to find that under under the influence of certain hormones, we're going to find that this primary oocyte will now undergo division, as we have talked about. And as that division has occurred, first it is going to be arrested. So it has entered already into meiosis 1. So as it enters into meiosis 1, it is going to reach in prophase 1. So as it reaches in that prophase 1 stage, it is going to be arrested, or it is going to be trapped. That is before birth, it is going to be trapped. So as it is trapped there, the whole division is basically taking place. But yeah, so as we were saying, you're going to find as that division multiplication is going on for the primordial germ cells forming this orgonia, it is just going to start going on to produce as much as ovum or egg cell that has to be produced before the baby is actually born. So you're going to find that before birth, these cells here, they are going to be arrested as they have entered in meiosis 1 at the stage that is prophase 1. They are going to be arrested. So at that point, these cells are going to be arrested, and they are only going to be released when this child is born and clicks puberty level. So at least starts clicking puberty. I believe that's somewhere 13, 14. Yeah, when it just enters teen, or the moment the same menstrual cycles start appearing, that's when these are going to be released. So as the child now clicks puberty there, it is now going, this ovum or this orgonium cell is going to be released, and it will enter into, it will continue its meiosis process. So as it enters, we are going to find that this primary oocyte is going to divide. It is going to undergo meiosis, in which it is going to divide to form the secondary oocyte and a polar body. These polar bodies are just bodies that are formed that are actually not even functional or useful for anything, but they just undergo death when they are formed. So as this primary oocyte undergoes division in that meiosis stage, when it is released from prophase 1 after the child clicks puberty, it is going to form the secondary oocyte. That secondary oocyte that is formed, it is then enters into the next phase, which is meiosis 2. So when it enters into meiosis 2, this one enters into meiosis 2, it is going to be arrested at metaphase 2 stage. So the reason why it is arrested at metaphase 2 stage is because this secondary oocyte, it is already matured. So what is needed just for it is someone to come and pay the bride price and make use of it. So what I mean by that is that what is just remaining at this stage is fertilization to occur. So I'm going to find as it reaches as it reaches metaphase 2, it is going to be arrested and it is going to be trapped. It is only going to be fertilized into a zygote in the presence of a sperm cell that has already also been undergone maturation and capacitation. That's what happens there. So it is going to be arrested in metaphase 2, and it is going to be trapped like that. Then, due to some hormonal surge that tends to occur, you're going to find that this is going to be ovulated. And when it is ovulated and you find that there is still no sperm cells to fertilize this egg, it is still going to be arrested in metaphase 2. It is only released in metaphase 2. Yeah, so it is going to only going to be released from metaphase 2, from this gel called metaphase 2, in the presence of someone who's going to bail her, in the presence of the spermatozoa to form a zygote. That's what happens here. So now, after knowing what happens to this one, and also before we go to the other part, we have to know that in the process when the fertilization occurs to form a zygote, so we are going to find that another polar body is going to be formed at this stage. When this is fertilized and the zygote is formed, another polar body is going to find that it is going to be surrounded by these cells, and these cells are known as the flat follicular cells, to form what we call the primordial follicle. So now what happens in this whole process of the primordial follicle coming in? So what happens is that the primordial follicle actually will start undergoing some development. And what I mean by some development is that in the presence of certain hormones like the follicle stimulating hormone, when it is present, it is going to start stimulating the growth of this follicle. So when this follicle starts growing, also the inside ovum, as we talked about it will start also growing, as we are seeing it inside here. It will start undergoing some certain developments, also growing in size and so on and so forth. So you're going to find that as this primordial follicle undergoes some growth, it is now going to grow, and it will be termed as the primary follicle. So this whole process of growth takes place in the influence or in the presence of hormone, the hormone that you mentioned, which is the follicle stimulating hormone. So what happens is that when it forms in the primary follicle here, you're going to find that this primordial follicle will start now growing. It will start further growing, or it will start growing furthermore. So you're going to find that it is going to be having a fluid-filled cavity that will start appearing, which is going to be filled with fluid and other components. And this fluid-filled cavity is what is referred to as the antrum. And as it is growing also, even the inside ovum also, as we talked about, it will start undergoing maturation, dividing, primary oocyte, secondary oocyte to start growing, and the ovum will start gaining certain characteristics or features, like the zona pellucida is also going to be there. Yeah. So we are going to find that there is also going to be the appearance of what we call the thicker cells, or rather there is going to be the presence of the thicker cells there that will be facilitating the production of a certain hormone that will be a hormone that is going to be converted into estrogen by the granulosa cells that are going to be present. So when this primary follicle divides and differentiates and forms the secondary follicle, which is known as also the growing follicle, this secondary follicle, as we have seen, its antrum or the fluid-filled cavity will enlarge such that it will even start pushing this ovum against the walls of the same layer itself. So as that happens, it is now going to mature into what we call the Graafian follicle, or the mature follicle. This Graafian follicle, it is already going to be ready in the presence of a certain hormone that is known as the luteinizing hormone, so that it can rupture. So what happens to the Graafian follicle is that the Graafian follicle is going to rupture when there is luteinizing hormone surge, or when there is the release of a hormone known as the luteinizing hormone. So that luteinizing hormone has got the ability to cause this Graafian follicle to rupture and release the ovum. So when this ovum is released, it is going to be released at this stage that we talked about. At this stage where it is arrested, that's where it is going to be released from. So as it is released, this whole process of this rupturing, releasing the ovum, is what we call ovulation. So when it is ovulated, they are released, and we say that there is a presence of these sperm cells to fertilize it, and fertilization occurs. What will happen is that Graafian follicle, when it ruptures and releases the ovum, it turns into a structure that is known as the corpus luteum. This corpus luteum has got the ability, or has got some functions in which it will start supporting the production of hormones, and these hormones are the ones that are going to be preparing the uterus, trying to maintain the integrity of the uterus, try to prepare the uterus for implantation as the fertilization has occurred. So when that happens, you're going to find that this corpus luteum will survive for about 3 to 4 months, until the placenta is formed, because when the placenta is formed is the one now that takes up the role of production of hormones that is produced by this corpus luteum. Then after that, this corpus luteum will degenerate and form what we call the corpus albicans. So in a condition where fertilization has not occurred, this also corpus luteum that does not stay for 3 to 4 months. It just takes about a few days or weeks, maybe let's say 14 days, when ovulation has occurred but there is no fertilization. So we're going to find that this corpus luteum will also die together with the same ovum, and it will be shed off, and you'll be seeing the same thing coming out as the message. So basically, these are some of the important points that we wanted to highlight on the whole process of oogenesis and spermatogenesis. Yeah, otherwise, if you have any questions, feel free to ask. Thank you very much for your patience and your time. Have a blessed evening.